Our next and final speaker is Jonathan Scolnick from Singapore, and he will talk about building a single-cell multi-omics toolkit. And he has been got a PhD at UC Berkeley and was at Dr. Sidney Brenner's at A-Star in 2009. And currently he's at the Department of Physiology at National University of Singapore to develop new assays. So Jonathan, please.
Great, thanks Jay, and thanks for inviting me to speak. A quick conflict of interest statement. So I'm a co-founder of Proteome Precision Medicine, which is commercializing a number of the assays I'm going to be talking about today. So my main interest really is how do you keep, or how do you treat a heterogeneous tumor like this in a way that you don't end up with a relapse in the end? And because we tend to basically exclusively work with human samples, the samples we get are very small and very precious. And so we actually spent the past couple of years developing some single cell techniques to try to extract the most information possible out of each of our samples. So we knew that we wanted to be able to look at protein expression in large part because there's really decades, 60 plus years of data developed by immunologists on single cell protein expression. And obviously in tumor biology, you're going to be dealing with a lot of immune cells. So this was an initial priority of ours. But of course, only looking at protein expression is problematic because you're biased in what you're measuring, right? You have to have, in a cytometry, you have to have an antibody to measure the protein of interest. So you have to know ahead of time what to look for, and so there's really no discovery mode. So when single cell RNA sequencing came out, that was this great boon because now we could collect thousands of data points per cell, and the data are relatively unbiased. And of course, we wanted to be able to collect both of those pieces of information to get the benefits of both the protein and the gene expression. But as we started doing analyses on various tumors, it was interesting that we found it actually was still difficult, even with the gene and the protein expression patterns, to be able to identify which is a tumor cell and which is a normal cell. So we needed to think about something a little more. We also, with these data sets, couldn't really look into tumor evolution. And we're missing some drug targets. So there are therapeutics that are available that target specific mutant proteins. And so that information wasn't available to us by just looking at the gene and protein expression. So more recently then we've added now mutation analysis. And the idea there again is to be able to really identify which cells are the tumor cells, be able to study the evolution of those tumor cells and get that additional drug targeting information. The major downside here is it really requires a lot of sequencing, but in the early research phase, I think that's okay and we can handle it. So here's the general workflow that we use. As I mentioned, we started out by combining protein and gene expression. We do that with these DNA barcoded antibodies. I'm sure you're all familiar with at this point. We use the 10x genomics system then to generate the single cell cDNA and these antibody derived tags. We use the cDNA then to detect mutations in a technique that we call single inversion targeted sequencing, or SIT-Seq. And then finally, obviously, there's a lot of data analysis that goes into this. And I'm going to talk about a particular type of analysis developed by our close collaborators, Sean Hoon and Winston Koh over at A-Star. So in normally thinking about a single cell RNA sequencing experiment, the first thing you would do obviously is collect your data, and then you would go cluster the cells, probably by Seurat. And then you would annotate the cells based on those clusters. And so in this case, maybe that gives you something like this, where you have a nice cluster of, say, CD4 T cells, a nice cluster of CD8 T cells, some NK cells, and then this cluster up here that kind of has a mixture of markers of T cells and NK cells. So for the purposes of this slide, we'll just kind of call them NKT cells. But we noticed with our protein expression that there was a bit of a problem. and that it didn't look like these clusters were actually forming correctly in order to identify the cell types of interest. So again, we worked with our collaborators, Winston and Sean, who developed a tool called MapCell. And what MapCell does is it annotates each and every single cell independent of the cluster that it landed in. And it does that by comparing it back to a database of cells that we've expertly annotated in the past. And what we see once we apply MapCell annotations is that our clusters are a bit mixed. So here we have CD4 and CD8 T cells mixed together, T cells and NK cells mixing throughout these clusters. And so you can see that's a very different answer you get compared to when we just annotated the cells based upon the cluster that they landed in. Now, of course, you could say, well, how do we know that this is correct? But here's where it's very useful that we also are measuring protein information, right? So this is an orthogonal measurement that we make on these same exact cells. So if we now take these same cells, cluster them by protein expression profile, even though we keep the annotation from MapCell, we can see that the cells cluster themselves out nicely. And this has sort of very real world consequences. So here's an example of a tumor we were looking at where now we've bioinformatically removed the tumor cells. So we're only looking at the tumor microenvironment. And if we were to use a standard cluster-based method for annotating our cells, you can see that we have 3 different cell types that we could identify. So basically there were 3 clusters. But when we applied MapCell instead, we find there are actually 13 different cell types. And again, of course, we can go in with the protein information and make sure that those cell types are correct. But obviously here, when we're looking at these tumors, we're going to be getting very different information and coming up with very different conclusions, whether we had 3 cell types or these 13 different cell types. Now, we don't just use the protein information to check that our annotations are correct. We actually feel that it adds an additional nice layer of information to our data sets. So here is an example. These were tumor infiltrating T cells. We could see that there were 8 clusters of T cells formed. And if we look at the 50 most differentially expressed proteins on the left and the 50 most differentially expressed genes on the right, what's interesting here is that only one of them matches. So CD8 is the only one in common between both. And so that gives us these 49 differentially expressed proteins that are very different from the top differentially expressed genes. Now, I'm going to switch over because I mentioned SIT-Seq. So I'm going to quickly run through how this works to do mutation analysis. As I said, we start with the single cell cDNA, which was derived in this case from the 10x system. We do a PCR amplification near our target of interest to amplify the target as well as the cell ID and the UMI. We then circularize that molecule and perform a second PCR to resolve the circle, which gets us our sequencing library. And we currently have a panel that covers about 80 mutations across these 16 genes. And that's something that we're continuing to build. Now, we don't take the entire panel and look at every tumor that way. The cost of the sequencing there would be really outrageous. But with the tumors we get, most of them already have some clinical sequencing data attached to it. And so then we can select mutations specifically to target in the cDNA for the single cell information. And so an example of that is this multiple myeloma patient. So the clinical sequencing had identified a Kras mutation with an allele frequency of 6.5%. That would roughly equate to 13% of the cells expressing, or the cells carrying the mutation. When we did SIT-Seq, we found actually 11.8% of the cells in our sample were expressing the mutation. So that was a nice correlation there. And what we're finding, which was interesting, but I guess we should have expected it, is that a lot of the mutations are appearing in multiple cell types. So we initially thought, well, our life would be easy if the tumor cells were the only cells that showed these mutations. But in fact, a lot of times we're seeing progenitor cells or stem cells are the place where the mutation occurs, giving you rise to different cell lineages that contain the mutation. Again, this shouldn't have been surprising because these mutations are going to be acting in a cell-specific context. And so that's how you get sort of lineage-specific tumors, even in the case where your mutation is occurring early on and in many different cell types. So again, we're now using these tools really to solve this old problem of how do you take this tumor, treat it such that you can treat all the cells so that you don't end up with this relapse in the end. So a number of people to acknowledge who contributed tremendously to this work. Jean Yeo is professor at UCSD, visiting professor here at NUS. Jie Hao is the technician in the lab who ran pretty much all the assays that I just talked about. Weiqi and Dominic, who developed SIT-Seq. Professor Qingwei Zhu at NUH and the Cancer Sciences Institute here, as well as his postdoc Jianbao and one of the clinicians in his group, Sanjay, who we have a very nice collaboration with. I mentioned Sean and Winston, who had developed MapCell, and Xiaojing and Eleni from Proteona have actually been involved in a lot of the analysis I showed. And finally, I have to thank our funders, the National Research Foundation and the Singapore MIT Alliance for Research and Technology. So with that, I'm happy to take any questions.
Thank you. Thank you, Jonathan, for your presentation.